everyone. Welcome to our Science at Cal lecture. Rosalind is with me. She is our public engagement specialist, and I'm the executive director of Science at Cal. Rosalind, where are people tuning in from?
Well, Dee, we've got a lot of Bay Area folks, of course. I actually saw a couple of people from the South Bay, the Peninsula, and I think the furthest someone is tuning in from is Florida.
Oh, Florida. Nice to see you. It's going to be late by the time we end for you, Florida. Well, that's great. I guess that this is a topic that spans the entire Bay Area, so it makes sense that we have a lot of Bay Area folks tuning in. We're so excited to introduce Karen Nelson, a professor in the Department of Civil and Environmental Engineering, on monitoring COVID-19 in the San Francisco Bay Area through Sewershed surveillance. Sewershed surveillance is quite a mouthful. I'm glad I only have to say it once or maybe twice. Before we get started, as usual, we are going to start with a land acknowledgement. We at Science at Cal recognize that Berkeley sits on the Hu-Chun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land has and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. So thank you for allowing me to take a moment to say that. I know, again, we're tuning in from all over the Bay Area, and this land acknowledgement applies to all of you. So again, at Science at Cal, you've probably joined us for other programs. If you're new to our programming, welcome. It's so exciting to have you. We host lectures. We really celebrate science, and we bring all of that wonder and excitement of the UC Berkeley STEM, that's science, technology, engineering, and mathematics, research to the community. All of our events are all of our events and programs, are free and geared towards public audiences. So I hope this is not the last time you join us. Please stick around. We have cafes, lectures, festivals. We are at Cal Day festivals. We are within the community, and we hope to see you live and in person in the fall. We are probably gonna, well, I shouldn't say probably, we are going to be back. I am making that declaration. We're going to be back in the fall, and we can't wait to see you with us face to face. We have campus community partners like the consulates, libraries. We work with, again, community members like First Fridays, all the farmers markets, community centers, artists. We are there, here, there, and everywhere in between. We have 2 more lectures coming up, the Science of Happiness. We have some Earth Science in August. These are our traditional lecture series. And then we have our Grounds for Science series. This is where we showcase graduate students across the university. And if you are part of our newsletter, you have already heard about these events, and you will be hearing more. If you are not, please join our newsletter at science@cal.berkeley.edu. We have a lecture series called Midday Science Cafe, where we team with Berkeley Lab. We have some interesting topics on negative emission technologies and the periodic table this summer. Our fall program will likely be debuted in the next few months, so stay tuned for that. We have a lot of fun things going on this summer, though. So many events for you to attend virtually. We want to thank all of our supporters. We want to thank people who donated today. We want to thank our founding council members, people across campus, the deans across every department, the vice chancellor for research who funds some of Science at Cal. And if you'd like to make a donation, please visit us again at science@cal.berkeley.edu. If you want to talk to me about how you can further support Science at Cal, please reach out to us on email. We are also on Twitter, Facebook, Instagram, you name it. And oh, that reminds me, this event will be on YouTube. So we will post this event. We will send it out across our listserv and across to all of you that RSVP'd today. So you will be getting this event in your inbox via video. The last thing I want to say is that we will be requesting, sorry if you can hear background noise, we will be requesting you to ask questions across, I should put my headphones on so you don't hear that. We ask that you ask questions along the entire presentation, all of Kara's presentation, and we will get to your questions at the end during our Q&A. So just enter those into the chat along the entire presentation. And also, if you'd like, we are also enabling you to be able to ask your questions, unmute yourself, but please wait until the end of the event to do that. I'm going to stop sharing my screen. Oops, that's not what I meant to do. There we go. If you click on reactions at the bottom of your screen and you click raise your hand, that's how you indicate to us that you would like to ask a question at the end of Kara's lecture during the Q&A. So please raise your hand. We will unmute you and we will call on you to ask your question. But again, if you just want us to ask the question for you, no problem. Go ahead and ask your questions in the Q&A. So without further ado, I'm going to introduce Dr. Kara Nelson, who is the Blum Chancellor's Chair of Development Engineering and a professor, in Civil and Environmental Engineering and Associate Dean of Equity and Inclusion in the College of Engineering at Berkeley. She teaches courses on innovation in the water sector, drinking water and wastewater treatment processes, pathogen detection, and inactivation. Oh, I don't know why that was hard for me to say. Taking into consideration the wide range of contexts that exist in low to high income countries. Her research program investigates practices for water reuse, disinfection, nutrient recovery, and international WASH, WASH, which stands for Water, Sanitation, and Hygiene, which draws on expertise in a wide range of treatment processes, including filtration, membrane processes, ion exchange, and disinfection with UV, sunlight, and ammonia. Dr. Nelson currently leads the engineering research thrust at ReNewIt, which stands for Reinventing Our Nation's Urban Water Infrastructure, and previously served on the expert panel advising the state of California on criteria to regulate indirect and direct potable water reuse. Very cool, Kara. Since April 2020, she has led a large multidisciplinary team developing innovative methods for monitoring wastewater as part of the COVID response and translating these tools to full-scale implementation. There's a website where you can learn more. Maybe she'll share that with you. It's covid-web.org. So again, without further ado, Kara, take it away. We're so excited to have you.
Thank you so much, Dion. I had no idea you were going to share all of that information in my bio, but thank you for that.
It was a lot.
Thorough introduction. And thank you for inviting me to be part of this Science at Cal program. It's such a treat to be here with this audience. I've been giving a lot of scientific talks, and it was really fun, or talks to scientific audiences, and it was kind of fun to put this together for a more general audience. I see some family and friends out there in the crowd, so thank you for showing up. And before I launch into things, I want to right away start off by acknowledging the amazing team that I have been working with on this project. So all of these folks listed here have contributed in small and big ways to the project I'm going to be talking about tonight. This team includes postdocs and PhD students, master's students, and undergraduates, and some full-time staff in our laboratory. And I want to specifically call your attention to Dr. Rose Cantor, Dr. Sasha Harris-Levitt, and Matt Metzger, who share the leadership of this project, and I absolutely could not be doing this without their contributions. So everything that I'm sharing today could not be done without this huge team. All right, so you saw a little preview of my next slide. What does poop have to do with this? Well, you heard a little bit in the introduction that what my research program focuses on is the safe management of human waste and wastewater. And that is pretty different than the perspective that most of you pretty have when you look at poop. When you think about poop, you may think of it as a cute emoji. If you have small children in your life or you are a small child, you might just think poop's great for making jokes. But generally, people think about poop as something that they don't want to deal with. You want to get rid of it. You want it to be someone else's problem. But when we look at poop, and I've been working in this research area for almost 30 years now, what we see is an incredible resource. And that is because there are useful things that we can get out of human waste. We can turn it back into energy or nutrients, or if we clean the water that it has contaminated, we can reuse that water for all kinds of important purposes. And in a drought-prone state like California, of course, that's incredibly important. In order for us to get useful things out of it, we also have to manage the dangerous substances that are in it, and that includes pathogens. And so that's one of the things that my group focuses on. But we learned when this pandemic started that we could leverage those skills and that type of research that we do in a whole new way and extract another resource from this human waste, and that is information. So that is what this project focuses on. And so what I'm going to be telling you about is how do we get useful information from this human waste and then leverage that and use that to make better public health decisions, and specifically with respect to the COVID pandemic. And the reason we can do that is because, like many pathogens that make humans sick, the virus that causes COVID-19, which is SARS-CoV-2, it actually infects the intestinal tract. So we think of it as an infection of the respiratory tract, but it turns out that the ACE2 receptors that you might have heard about where the virus binds, so these are the virus binding sites here, they can actually attach to the same sites in our intestines and infect our intestines. And so then they are excreted by people that are infected by SARS-CoV-2. And then we can measure that in the wastewater. Now, one thing that you might also be worried about is, oh, well, what happens to all of that wastewater? Well, fortunately, because of the design of SARS-CoV-2, because of its structure, there's a lot of evidence now that it is very easily removed and inactivated by our standard wastewater treatment processes. In fact, it may not even survive very long in our sewers. And that's because the spike protein here is actually very easy to damage. And this whole pink coating that it has is made out of lipids, which are also easily damaged by substances in our wastewater. So there's consensus now that there's no substantial risk to workers that work in our, to manage this waste for us, or of this virus being discharged into the environment. But we can still, in our wastewater, find the RNA. So even if parts of the virus on the outside are being damaged, the signal that we look for to detect it in our wastewater is this ribbon-like substance in the middle, the nucleic acids, which in this case are RNA, not DNA. And so if we can detect that signal, then we can get information from our wastewater about about the occurrence of COVID infections in whoever has produced that wastewater. So this field, what I'm going to be talking about today is this application of wastewater-based epidemiology. And that is a whole field that has just exploded because of this pandemic. It existed before the pandemic, but wasn't being used for many applications. So what this consists of is measuring things like COVID in our wastewater. So imagine that we can collect a sample here in the sewer pipe that's leaving this building. If we detect SARS-CoV-2 in that wastewater, that tells us that at least one person, maybe multiple people in that building are infected with COVID. And we can use this information to inform the field of epidemiology, which is the study of the occurrence of illness and how illness is moving in a population. And then we can use that to inform response and then also measure whether interventions are actually working. So we can measure we can collect wastewater samples at individual buildings, we could collect them in neighborhoods, and we can collect them at the entrance to treatment plants. And this is an incredibly efficient way to get information about the occurrence of SARS-CoV-2 in hundreds of people up to hundreds of thousands or even millions of people in a single sample. So it's incredibly efficient. And it also has some other advantages. So in addition to being an inherently pooled sample, imagine how difficult it would be to go around and collect a nasal swab or a saliva sample from all those people on a regular basis. So it's very efficient. We get information on a lot of people, but it also has the potential to be less biased than the other types of public health information that we are using for making decisions. So clinical case data, as you can see in this graph on the right, the different colors represent the testing per capita in the city of San Francisco, and you can see that it varies dramatically from neighborhood to neighborhood. So depending on how many tests are being administered, we will learn about the infection prevalence in that community, but it's not distributed evenly. And we know that some communities have a harder time getting access to testing or are more reluctant to getting testing. And then as we move into this later phase of the pandemic, less money and resources are being spent on doing tested. So right now in most places, you can only get a test if you're symptomatic. We're not doing surveillance testing anymore. So testing is biased, whereas the wastewater information doesn't suffer from those same biases. And there's one more potential advantage, which is that there's evidence that the shedding from the fecal tract actually starts very early in an infection. So we may be able to pick up the signal from that infection sooner than it would be detected through a clinical test, because many people will only seek that clinical testing once they show symptoms, which is actually after the infection has progressed quite a bit. So many groups realize this potential early in the infection or early in the pandemic. And furthermore, information started coming out early in the pandemic about the disproportionate burden that some communities were experiencing. So communities of color, lower income communities have shouldered a much higher fraction of the illnesses and the deaths in our communities. And so one of the ways in which this less biased information from wastewater can potentially help is to better target resources for reducing this disproportionate burden in some of our communities. Okay, so another headline. I want to take us all the way back now to the very beginning. For us at UC Berkeley and in the Bay Area, things got real way back on March 17th when the Bay Area, all 6 counties issued a stay-at-home order, and UC Berkeley was also closed. And when I went back to look this up, I was reminded that initially the order was through April 7th, 2020. So we had no idea, right, what we were heading into. But everything in my laboratory, all of our research was put on hold. We have an experimental lab and we couldn't go there. So we started thinking, what can we do? And realizing that these skills that we had in our research group could potentially be very useful for providing information as part of the pandemic response. But this was an opportunity unlike any other, because usually when we do research, we spend years conducting the research before we actually make any recommendations and try to act on it. But we didn't know how long this pandemic was going to last, and it still continues to feel incredibly urgent. And so we did something very unusual, which is we started conducting research to develop the methods to measure this virus in wastewater at the same time as we started putting into place the infrastructure that we would need to actually launch a program to conduct these measurements and produce useful data in the Bay Area in real time. This is not something I recommend. I generally recommend research and then implementation, or at least wait a little longer, but we didn't feel like we really had that flexibility because of the urgency. So one of the benefits of doing all of this in tandem is that we have been learning very quickly how to provide, like what type of information we can provide and how to provide this information that is most useful for informing the public health response. So I'm going to walk you through a story of some of the research that we've done, and then also how we've implemented this COVID web program. Before I launch into the story, I want to give you a sense for how this information can and has been useful. So I'll give you the can, and then when I get to results, I'll talk about some specific examples. But back to that original idea of wastewater-based epidemiology, depending on where we sample, we can potentially inform different types of public health decisions. So if we're sampling at the building level, this can help people in decision-making positions decide, okay, we need to go and test everyone in that building because the wastewater was positive, so there appears to be at least one infected person, and then we're going to do contact tracing and isolate that so we can get a quick response and also confirm any clinical data that already exist, whether positive or negative. If we sample at a slightly larger region, maybe a neighborhood or section of a city, then we can start to see trends across neighborhoods, identify hotspots. Again, this can help to deploy testing capabilities or supportive public health interventions, helping people figure out how they can manage sheltering in place, affording being able to miss work. We can use this to motivate people to take things seriously and to get tested. And then finally, if we sample at the largest level, at the treatment plant level, and we have a few sampling locations in the Bay Area that capture the waste from hundreds of thousands of people, we can use this information to inform decisions that are being made about whether it's safe to relax shelter-in-place restrictions, or if we're seeing an increase, that we may need to actually make them stricter. What's nice right now, we're in the part of the pandemic where we have very few infections occurring, and the wastewater is a very efficient way to confirm that infections are remaining low. So here's a rough timeline and this is what I'm going to walk you through. So I'm going to walk you all the way back to when we started collecting samples and developing our methods, and then how we moved from there to establishing our testing laboratory, and then launching a full-scale monitoring program early last fall. I'll share some results with you, and then give you share some thoughts on what might happen next, both in terms of how we can use this resource to continue to manage SARS-CoV-2, and then what this field of wastewater-based epidemiology might hold for the future once COVID is no longer our primary concern. Okay, so methods. So when this whole mess started, nobody in the world had a method for measuring this virus in wastewater because it had not existed previously. And we were fortunate to have a collaboration with some students in another laboratory, especially Oscar Whitney, who had never worked with wastewater before, but had a lot of experience working with nucleic acids in different types of matrices. And we recognized that there were some unique things about the SARS-CoV-2 virus that we might be able to take advantage of and develop a different approach to detecting it in wastewater. So most methods that existed for detecting other wastewater or other pathogens in wastewater, other viruses in wastewater before this pandemic, focused on detecting the virus itself. But we recognize that a lot of this virus might be breaking open, like I explained earlier, because it's a pretty wimpy virus. So we wanted to develop a method that would allow us to detect not just intact viruses, but the free RNA. And so the method that we developed is, it's pretty neat and it's pretty cheap. And the first thing that happens is we add a bunch of salt to the wastewater to intentionally break up the virus particles and release their RNA. So we actually send out the tubes to our partners with salt already in them. So when we get our tubes, and this is what it looks like when we receive the wastewater in the lab, the viruses are already lysing and releasing their RNA. We heat them to make sure that lysis is complete and to make the sample safer to work with, we do a coarse filtration, and then we add ethanol to make that RNA sticky. And then we pass the samples through these columns, and way down here where you can't really see, there are these silica beads that the RNA sticks to. So we pass the whole wastewater sample through the column, and what we're left with is the RNA sticking on the silica beads, and then we rinse that off and collect a very small volume, only 200 microliters, in a small microcentrifuge tube, and that's what we use to measure the RNA. And we use that using a pretty conventional approach, quantitative reverse transcription, PCR. And in that process, we make a DNA copy of the RNA, and then we amplify it billions of times so that we can detect it. And as you'll see later, this detection method is incredibly sensitive. Actually, I'll go ahead and tell you right now, since we're on this, we think that our detection limit is probably sensitive. Our method is probably sensitive enough to detect a single COVID infection in about 100,000 people. So this method is incredibly sensitive. Okay, so yeah, that took a while. We did this really fast, but we developed a new method and we validated it. And then kind of in parallel, we started building out the lab capacity in order to be able to process a lot of samples. So because SARS-CoV-2 is in wastewater, we can't be sure it's not infectious. We have to take a lot of biosafety precautions. So we actually had to identify a whole new space on campus because my regular research lab wasn't big enough. So we identified a whole new space over in the College of Chemistry and built it out with all the right biosafety precautions, special biosafety cabinets, and there's a lot of approvals that are needed, and then started hiring full-time staff to work here. So here is our incredible team that helped to launch the initial lab, and I want to call your attention to Matt Metzger here, who's our lab manager, and Melissa Thornton, who was our first lab tech, and they have been working full-time with us since early last fall, processing hundreds of samples a week in we call it our pop-up laboratory because we don't expect it to stick around forever, but it's already been operational much longer than we expected. And then the other members shown here are members of my research group who completely pivoted off of their regular research projects to help us launch this effort. So again, in parallel, we started reaching out to partners across the Bay Area that would be interested in collecting wastewater samples for us. And some people started coming to us once they started hearing on the news and talking to others about the potential for wastewater to be so useful. So here's the website that Dion mentioned, where you can learn more about our regional monitoring program in the Bay Area, which is called COVID Web, or COVID Wastewater Epidemiology for the Bay Area. And here's an overview of what we do. So we sample at 37 sites, shown by all of these orange stars here. About 37 sites where we're sampling 1 to 5 times a week now. We have processed over 3,500 samples to date, and we get all those samples from 19 different wastewater agencies, from the UC Berkeley campus, and from a number of residential facilities that have very vulnerable populations living in them. And then we report all this information out to the different county and city public health agencies that cover the jurisdictions where the wastewater is from. We also report this information and talk frequently with the California Department of Public Health and their counterpart on the water side, the California Water Board. That's sort of who's been given responsibility to oversee these efforts at the state level. And then the Center for Disease Control has launched a national effort, which I'll touch back on later. And so we're participating in a pilot where the information gets fed all the way up into a CDC data collection system called NEWS, which is the National Wastewater Surveillance System. And a quick overview of how all these pieces fit together. So again, we collect samples at these different scales. And the type of sample we collect is a 24-hour composite. And our wastewater agency partners have been very generous in using equipment that they already had to collect these samples, and in some cases installing them in new locations. So a 24-hour composite sampler automatically takes an aliquot of wastewater from the sewer every, periodically. I think the most frequent can be about every 7.5 minutes. And so when we test a sample, it should be representative of all the waste that has been deposited over a 24-hour period. And so here's some of our dedicated team collecting one of our samples on the UC Berkeley campus. And so those samples get sent back to us in the lab. We add a positive control, another virus at a known quantity, so we can track that all the way through. We do our 4S method, and then we quantify the SARS-CoV-2 virus using that PCR reaction. We quantify another virus that we always expect to be there at constant concentrations, and then we measure our positive control. And then we do a whole bunch of quality assurance and quality control, and that what we end up with are concentrations that we can track over time and over space. All right, now I've brought you up to speed enough that you can appreciate some of the results that we are able to generate. So I'm going to walk you through some results that we have from monitoring the wastewater across the Bay Area, some really interesting results from one of our residential facilities, and then some results from our campus. So this is one way of capturing most of the information that is in those 3,500 samples that we've processed to date. So what we're looking at here is, these are our different sampling sites. Each row represents a different location, and then time across the x-axis. So starting back in August of 2020 up through, this is last week, so June of this year. And the colors represent the concentration on a log scale. So our detection limit is somewhere down around here, and we do have some sites where we're at our detection limit. So that's about 4 gene copies of this virus per milliliter of wastewater. And then you can see we had some samples that were the concentrations were so high, they were in several thousands of copies. So that's the range of concentrations that we've seen over the course of this whole pandemic. And so when you just look, when you take in this whole heat map, what you can see are some very interesting trends. If you look at it from left to right, you can see that back in the fall, when we started sampling, the concentrations were lower in most of the locations, although there were some hotspots. And then as we moved into the like the Thanksgiving and winter holiday season, that's when we had our big surge in the Bay Area and across California. And you can see the whole heat map just lighting up with high concentrations. So, infections going way up everywhere. And then what's very encouraging is you can see in general, the colors getting darker as we move to the right. And so concentrations have come down everywhere to some extent, although we still have some hotspots. So here's one site where we're still measuring fairly high concentrations. Here's another one. Here's one where we had sort of like a little mini outbreak that was fortunately short-lived. So we're still seeing a lot of differences across different communities across the Bay Area. And this information can be really useful to our partners in public health in trying to track down outbreaks, early before they really spread, and isolate those infections. And then here's an example of a specific time series. We generate these graphs for every one of these rows. So for every location, our partners in public health have access to a dashboard where they can see any of this, all this information is right at their fingertips. And they can also see a more detailed graph of the concentrations over time. So on this top graph, you're seeing the time series of wastewater starting back in September in one of our cities at 3 different locations. So in this city, there are 2 different wastewater treatment plants. And then we also have a sampling site that's just a specific neighborhood that's nested in the service area or the sewer shed of wastewater treatment plant 2. And then on the bottom, where we've paired this data with the clinical case data. So this is number of positive cases, daily positive cases per 100,000 people. So a few takeaway points. One, the wastewater data and the clinical case data agree with each other to a large extent. Neither one of them represents the perfect truth, because we know that the case data also has some biases. On the wastewater side, the samples aren't perfect either. Sometimes the samplers clog. Sometimes they're not well mixed. Something might go wrong in the laboratory, although we track that very closely. But you can see that the same general trends hold. And we can also see that the number of infections per capita was, well, in the case data, we can see that it was higher in this neighborhood. So this is a neighborhood that's experiencing a much higher COVID burden than other surrounding neighborhoods. And if we were only sampling at wastewater treatment plant 2, which is in red, we wouldn't have noticed that. So sampling at these smaller scales can really help us identify communities that are suffering more and better direct resources to them. And so we see that trend in the case data. We also see it in the wastewater. And we even see it persisting, although fortunately the cases are finally coming down. They're still much higher than in the area served by this green treatment plant. And as the number of infections, or as the number of tests being administered really goes down, and sort of in the portion of the pandemic that we're in now, we're expecting that the wastewater data will be even more valuable, because our partners in public health are increasingly concerned that they're missing infections that are out there, especially among people that are asymptomatic. So if you don't ever get symptoms, you're never going to go and get tested. And so that may not be discovered until you've already infected other people. With the wastewater data, hopefully we can catch that earlier. So this city where this wastewater data is being collected from was in the yellow tier before we got rid of the tier system on June 15th. So many of our communities around the Bay Area were in the yellow tier. And one thing that's very encouraging about our method is that we are still able to detect the virus in the wastewater, even though the number of new daily cases is very, very low. So to be in the yellow tier, you had to have less than 2 daily new cases per 100,000 people. And it seems that our wastewater methods are sensitive enough to distinguish that. So we think that this is going to be a very useful tool for providing reassurance that it's still safe to, we've just reopened everything, and that appears to be safe. If we do start to see an increase in infections, we should be able to detect that in the wastewater, even if people aren't getting tested clinically or there are asymptomatic infections. Okay, the next thing I wanted to share with you were some results from a high-risk residential facility. So on this graph, we flipped the order of things now. So now on the bottom, we're seeing the wastewater data. And then in the middle panel, the new cases that are detected among residents. So when the sampling first started, this was back in early December, no positive clinical cases, no signal in the wastewater. But lo and behold, on this day, we detected a positive signal in the wastewater, but there were still no clinical tests that had come back positive. But unfortunately, right after this, they started getting some positives in the individual testing that they started doing. And we saw the wastewater concentrations just skyrocket. These are some of the highest concentrations we've ever measured. So this was a horrible outbreak. But the medical officer of this facility has shared with us that she feels like the wastewater information was very helpful for her to control the outbreak, and that without this information, it might have been even worse, because the fact that the wastewater concentrations kept going up helped her make decisions to continue to impose even stricter shelter-in-place restrictions. So really putting everyone pretty much on lockdown, which she was very reluctant to do. if things weren't so severe. So this information was very useful in that case. And then what's been really reassuring is this is a time series now looking out further. This is looking all the way up until last week. For the last 4 months, the wastewater has been no detection of the virus except for a few small, three samples. And so this has also been very reassuring because they've continued to allow people to have more ability to circulate without the fear of transmission. And at this point, almost everyone is vaccinated. So they're not expecting any infections, but the wastewater, again, provides very reassuring information. And once in a while, they have an infection among a staff member. They want to make sure it's not spreading to the residents. So the wastewater can help ensure that. And then finally, what's going on at UC Berkeley? So UC Berkeley did not have a lot of residents in campus living, campus residential buildings this year because we were doing almost all virtual school. And in the dormitories, they were testing everyone twice a week, doing nasal swabbing. So the wastewater didn't have a lot of potential to add more information. But we did identify some priority locations for where wastewater testing could provide information. One of them was in the student family housing, where there weren't resources to do regular swabbing of all the residents. And then we also have one over in Clark Kerr, one of our residential facilities where they weren't doing regular swabbing. And then a few manholes that are downstream of where there's a lot of student residences. And so we started regular sampling in partnership with our environmental health and safety team here on campus of these locations. And this is information that's actually posted on the public dashboard. Now the bad news, well, okay, more on how this information is used by campus. So whenever there's a positive, campus would use this information to send out a warning to the residents of that facility to say, the wastewater is positive, there's COVID, please go and get tested so we can track this down. The bad news, so if you on the website, and this is on the regular campus dashboard, if you hover over one of these locations, then the testing data comes up. The bad news is that last Friday there was a positive in the University Village wastewater. So hopefully, we're awaiting the new samples from this week, but hopefully that signal will disappear. And they are tracking down that infection or infections right now. Looking towards next year, campus is most likely going to be using wastewater monitoring to be keeping track of what's going on in our dormitory facility. So we're identifying some new sample locations because those will be fully populated again. Okay, so what is next? In addition to continuing to sample on campus and now focusing on the dormitories, most of our partners in the Bay Area have indicated that they would like us to continue this sampling. So we are planning to keep our laboratory open for at least another year. At the same time, we are also supporting the California Department of Public Health is in the process of building out a laboratory of its own so that they could take on this role of providing monitoring as a service to our public health agencies. And so we're working with them closely, and they're also working with a few of the other academic groups in California that have been doing this and have a lot of experience to share. And that's because there's a lot of evidence now that this information is very useful. So I've mentioned that one of the things is confirming that cases remain low. And we also want to have, we want to use all this infrastructure that we've put in place to get an early warning if cases do start to increase. And we hope that never happens, but where vaccination rates are lower, that's still a concern. And then we really hope this doesn't happen, but what if a new, more contagious variant does arise? Or what if a variant arises and starts circulating that the vaccines don't provide protection against? So all of this ability to do this efficient monitoring can really help us to get an early warning if any of these things should happen to occur. And like I mentioned earlier, the CDC has signaled that it plans to fully build out this national wastewater surveillance system as a national resource for collecting all this information that is coming in from wastewater monitoring programs. There are also other ways in which wastewater testing could be useful that are not fully developed yet. And one of the areas that we're working on is the ability to track specific mutations and variants and their evolution in our populations. And so I wanted to share just a tiny bit of background on that because it's not an easy thing to do. So here's an example of a genome from an important variant. So here's the annotated genome of SARS-CoV-2. It has 30,000 different base pairs in its RNA. And when mutations occur, in particular in this spike region, those are the ones that seem to have clinical significance because they affect the ability of the virus to bind to its receptor cells, and how strong that binding is, and also the ability of, or whether or not that variant is affected by antibodies, either as therapy or as part of our immune systems, or that we acquired through vaccines. So we're particularly concerned about mutations over here. But what's complicated is that, so this is just B117, has this family of variants has about 17 different mutations, and you can see they're spread throughout the genome. And when we sequence wastewater, we're not getting information from just a single person, from a single variant. So a single person should be infected by just one variant, but when you collect the wastewater from a whole bunch of people, you could have multiple variants present. And the way that we do the sequencing, it requires chopping up the genome into different pieces, and then it's very difficult to put them back together again. So we just get little snapshots of the genome in which mutations are present, and we don't necessarily know which ones are paired with other mutations. So we're trying to develop some new methods to be able to detect the variants. So when there's only one mutation, we call that a single nucleotide variant, a SNV. And so when we get our sequence data, we can graph the different locations of the mutation. So this is like the 30,000 base pairs across the bottom. And we can look for specific mutations and specific variants. And then in each sample, so each row represents a sample, we can look at the relative abundance of that mutation. And then when we start to put the pieces together, we can make predictions about which variants are most common in a particular sample. So here's an example where this sample seemed to be dominated by one of the California variants, because we see all these cells lighting up yellow. And this right here is the L452 mutation, which is of clinical significance in the spike protein. So that's the work that we're doing right now. This is led by Dr. Rose Cantor in my group, who's a bioinformatician and that has really deep expertise on these methods. And one other potential that this ability to sequence wastewater has is, it's complementary to clinical data, but it also might have the ability to help us detect variants before we see them in clinical samples. So here's some early data that we published last fall, where these are different samples in the rows, and again, the different positions in the RNA across the bottom. And wherever you see a bar, that's a mutation that was detected. The mutations shown in red, those matched sequences of samples that have come from clinical patients in California. But the ones in gray and blue had not yet been reported in patients in California. And that indicates that we were picking up mutations that had been introduced to the Bay Area. And we could see that in the wastewater, but they hadn't shown up yet in the clinical samples. So we're optimistic that we might be able to get early warning of new mutations, new variants when they show up in our region. right? So that's one thing we're very actively working on right now. And then looking even further into the future, we've started looking for other pathogens, and there's potential to look for other health markers in wastewater. There's potential to look for antibiotic resistance genes. And then another application near and dear to my heart, taking us back to our regular research area, which is in California. To address the effects of drought, we are starting to think about, or starting to work on, turning wastewater back into drinking water. And so having a really good quantitative characterization of the pathogens that are present in our wastewater can help us ensure that the drinking water we produce from it is extremely safe. And just a little snapshot into some of the other pathogens that we're starting to look for. These are time series from 2 different locations in the Bay Area. And you can see that some of these viruses are present in pretty consistent concentrations, but Norovirus, which is the famous, before COVID, it was the famous cruise ship disease that would give you projectile vomiting. And you can see that the concentrations, they exhibit the pattern of outbreaks. So the concentrations go up and then they come back down and then they go up again. And same for Bocavirus, which is an interesting virus that is primarily transmitted in children. So this may be interesting for understanding the transmission of pathogens in our school-age children, depending on different practices that are being used to reduce transmission in our schools. And then finally, looking really much further ahead in terms of preparing ourselves for a future pandemic or biosecurity, we are starting to work on methods that will allow us to detect unknown viruses. So instead of looking for things that we expect to be there, like SARS-CoV-2 or Norovirus, looking for unknown viruses through sequencing. And CDC also is recognizing these potential application areas, from emergency response to pandemic preparedness, identifying emerging infections, and bioterrorism. And people all around the world are working on this. So we are one group, but there are literally thousands of researchers working in this area now, all distributed all across the world. And so I think wastewater-based epidemiology is here to stay. My last slide before thank yous is just a kudos to our whole team. We were very honored to receive the UC Berkeley Chancellor's Public Service Award last month for our partnership with our Bay Area partners. So here's our team celebrating in front of the Campanile last month. And I will finish with thanking all of the partners that we work with and our public health agencies and the wastewater agencies that have been so dedicated in providing us samples, and then acknowledging all of our funders who saw the potential for this new tool to be developed and had faith in us and invested in this project. So I want to make sure to acknowledge them. And thank you all for your attention. And I will stop sharing my slides and we can move into Q&A.
That was wonderful. Thank you, Kara. A round of applause from everyone, since you can't hear us. If anybody has a question that you would like to ask Kara yourself, please go ahead and click the reactions button and click the raise hand, and we will call on you. Oh, Tom Klein is already with us. Tom, you want to go ahead and ask your question?
Yeah, I'm wondering, in light of the value of this whole wastewater thing, obviously you're going to continue sampling while the pandemic's around. But is there any plan to have a permanent library of wastewater samples in the future? For example, I don't know if anybody was collecting and keeping wastewater so we could tell when this whole thing actually started in the Bay Area. But going into the future, wouldn't it be a good idea to preserve samples, maybe with some fixative or something, permanently every day into the future, so you could go back in a library and once you saw something and see when it started? Are there plans?
Yes, there are definitely people working on that. We have not been specifically focusing on developing a wastewater archive ourselves. There are some very interesting ethical implications that come up when doing that, because it's not clear whether one would need consent in order to collect a sample and store it and use it for a purpose other than what was originally intended for. So like with our partners right now, we tell them, you know, our intention is to use these samples for looking for SARS-CoV-2 and some other pathogens. But we haven't said, you know, we're going to go look at what type of medicines people have been taking and we could find in the wastewater. So,
but they're anonymous.
They are, yes, they are anonymous, but there are still some important ethical considerations about how information could potentially be used that I think are really important to think through. And there's a lot of people, there are groups starting to think about that. But there are other groups that are specifically focused on providing archival material like you suggested. So thinking about these samples more as a type of observatory, and I think that is likely to happen, yes, and probably even at the state level. And we do actually have access to some samples that were collected, wastewater samples that were collected before we know, or before the first detection of a case in the Bay Area, that we are going to be analyzing to see if we can better pinpoint when COVID showed up in California.
Great, thanks a lot.
Yeah, that was a great question. Before we get to the next one, there was a question in the chat that asked about the length, the viability of the sample. And it sounds, since we're on this topic, it sounds like the virus remains in the sample for an indefinite amount of time. What would you say?
Yeah, so it depends on how the sample is handled. If you just keep wastewater around at room temperature and don't do anything with it, the SARS-CoV-2 virus is going to probably disappear within a few days, even the RNA. So it's actually not very stable. But if you throw that sample in a -80-degree freezer, like we do, then it will probably stick around for, like, the signal will probably go down, but you could probably still detect it, you know, at least a year later. And then if you preserve the RNA, if you extract the RNA and preserve that at -80 degrees, we think that will probably last for a couple of years.
Got it. So even if testing did continue, there'd be resources, not just about the testing, but creating facilities that could handle that historical data. Great. So let's hand things over to Grace. Grace, do you want to go ahead and unmute yourself?
Yeah, thank you so much. That was, as someone who doesn't know that much about wastewater epidemiology at all, it was very insightful and I feel very well informed. So thank you so much for such an insightful presentation. I definitely am interested in the social side of this. And, you know, as we all know, COVID is such a polarizing issue, and our ideologies and our knowledge changes on a day-to-day basis. It's, you know, very time sensitive. And so I'm wondering how you've seen the balance of understanding how we can help manage like communities' prejudices and our personal opinions, especially in some states where COVID is taken less seriously. And then like the need for like true public health initiatives that work to benefit the community.
Wow, that's a really good question and very open-ended. So I think one way to start thinking about the sort of broad concerns you've raised is that the wastewater information does provide a different entry point for having conversations with communities and working with community-based organizations to understand health, better understand the health status in those communities. And the fact that we are now, so a whole new, many new types of relationships have come out of these efforts. So in the past, you know, in the before times, there weren't that many reasons when wastewater agencies and sort of the wastewater sector had to talk directly to the public health departments and vice versa. And so through these efforts, we have developed whole new relationships that are going to persist beyond the pandemic. And then there are a lot of really amazing and committed people in public health that work very closely with community partners to try to improve health in communities and build trust and build relationships with community members to improve health and improve the acceptance of different types of ways that that community could be supported. So I don't know if that's getting at your question. You might have also alluded to, people that are sort of skeptics about COVID and like, oh, it's not that serious, or, I'm not going to get vaccinated because I don't think it's going to affect me. I think that's a bigger challenge, and I'm not sure that wastewater has a unique advantage in terms of addressing that, unless people are already accepting of data, data and analysis and sort of the scientific approach. Because that's a big challenge, right? If people are not accepting of the scientific approach and how to interpret, scientific results.
Great, thank you. Thank you, Grace, for that great question. So let's open it up to Dan Fishman.
Yes, it was a wonderful presentation, very clear. I'm intrigued by your comment about reusing wastewater in the context of the drought. How would you apply this technology to facilitating the process of reusing wastewater?
Oh, this is one of my favorite topics. So we already actually do turn wastewater back into drinking water in California, but we don't do it directly yet. So right now we can put our wastewater through primary, secondary, tertiary, and advanced treatment, and then put it back in the ground and reuse it for drinking water. And we already have regulations to do that. And the next step is putting it directly into a reservoir. And San Diego is moving forward with the first project to do that. And then the final step will be taking that advanced treated water and putting it right back into our drinking water pipes. And that's what we call direct potable reuse. And we have all the technology to do this, but what we need to know with even more confidence still is exactly how much treatment do we need to get from the wastewater to the drinking water, so that the risk of anyone getting an infection is so low that it would be, we can't even quantify it. So by just gathering a lot more data and measuring more pathogens and more wastewaters more frequently, we're going to get that ideal data set that we need to do all those engineering and risk assessment calculations that tell us exactly how much treatment is needed. And there's a bit of irony in the position that California is actually in right now, which is that some people actually feel like the regulations that are moving forward for this practice are actually overly conservative. We're requiring that so much, we're requiring more treatment between here and there than we actually need to. And the reason is because we don't have a good enough characterization of the pathogen concentrations in the wastewater, because not that many people have been worried, been working on it and worried about it. So it's possible that one of the outcomes of better information will actually be to slightly, reduce the number of barriers that we need to put in place, which will make the treatment more affordable and more accessible to more Californians. So that could be a good outcome in the end.
Great, thank you both so much for that question and answer. Really good question, Dan, thanks for asking. Kara, we did get a couple of questions in the chat regarding the turnaround time for these test results, and also getting that info, to public health officials in order to make decisions.
Yeah, those are excellent questions. So we can get test results out in a day, or like from morning to evening. So if we get samples in our lab before 10 a.m., we can get the results out by that evening, no problem. If we get them before noon, We might be able to if everything goes right and our lab staff's willing to stick around. So like for our campus sampling program in the fall, we are going to be aiming to get same day results. And so we're hoping all of our samples will come in before 10 a.m. And then as soon as our results are, as soon as we've gone through the QA/QC, they're even right now, they're posted on our data dashboard that all of our public health partners have access to. So they can see the results immediately as soon as we have them.
I know there's been a lot of, there's still a lot of research going on in how COVID is presented in the body, but there was the idea that people were getting COVID twice, or were they showing COVID, like a second surge of COVID in their body? Does the fecal matter show that second surge that sometimes we're seeing? Is it more sensitive to than the nasal swab? How does that compare in sensitivity?
I wish I knew. So, so here's an area where there's not enough research. So there's not a lot of research groups that are going around and collecting stool samples from folks in that situation, who are potentially experiencing a second infection. So most of the secondary infections, are likely asymptomatic. And so people don't even know they have them. So you wouldn't even know to collect their stool to measure it. If you do get a symptomatic infection, somebody may be collecting these data now, but I haven't seen it yet. So unfortunately, we've got some data gaps. And another one is even just how early in the infection cycle fecal shedding starts, because nobody's out there collecting stool samples before you get symptoms. So all the fecal shedding data we have is from people that have already developed symptoms.
Yeah, and I was wondering, we had some questions actually about the distribution of sites, and then you showed people were asking questions, then you'd answer the question later on. You were so good at that. You were like fabulous at answering questions that we hadn't asked. But as it relates to the distribution, are there more testing sites, say in urban areas or areas where there's universities present, where research is actually taking place? Or are these happening at a local level or a local government level?
Right. So the entire Bay Area is pretty much covered through our project. And then my colleague, Ali Baim at Stanford also has a monitoring project that covers much of the South Bay. And so the Bay Area itself is pretty well covered and certainly has benefited from the fact that both UC Berkeley and Stanford are here. Davis is also really well covered because they've got UC Davis. But this is not the case across the whole state. And so there are a number of groups that are working to try to expand wastewater testing to communities that haven't benefited from it yet, in particular disadvantaged communities in the Central Valley, and smaller communities in general that haven't benefited from being part of one of these university-led initiatives. And it is something that CDPH is definitely taking into consideration as they think about how to keep this work going at the state level.
Great, thank you. Perfect. In that heat map that you showed when there was no data, did that mean that there was no virus or there wasn't any data collected at that site?
Oh, for the campus data, yeah, you should go on with, well, actually, I'm not sure if that question was about campus or the
heat map, how some of the boxes were white.
Oh yeah, the white boxes on the heat map, we have no sample for. So over the life of the project, there are some sites we've retired, either because they weren't interesting or it was no longer feasible to collect samples there, or in some cases we've had the operators who collect the samples get sick and are out on leave, and so there's no one to collect the samples. There were some challenges around the winter holidays, of course, getting samples collected. So
okay, so no data as opposed to no COVID.
Yeah, there are literally like 6 samples maybe all within the last month where we have detected no COVID.
Wow. Okay, that's the statistic. Okay, yeah. So it's not, wow, interesting.
So there are a few, none of our large sampling sites where we have like more than 100,000 people have we gotten a non-detect yet. And so the only places we've gotten a non-detect are in smaller communities that have somewhere between 8,000 and maybe 40,000 people. And that seems to agree with the clinical case data. Like there literally were no COVID infections for a few days reported in that community. But yeah, I mean, it is kind of a wake-up call if you were wondering, you know, is COVID still out there? It's still out there everywhere that we're measuring it. Even the places where it's been negative, none of them have been negative consistently. So it's everywhere still in low numbers.
Good reminder. Thank you.
Thanks so much, Kara. Before we close out for the evening, I did just want to ask kind of an interesting question. I know that, you know, probably collecting these samples was probably tedious and laborious. And so someone asked, is anyone looking, you know, at testing robots at these locations to kind of help lessen that labor in getting the samples?
Yes, definitely. In fact, in the private sector, sort of all over that. So I'm expecting a lot of companies are going to come up with new and improved sample collection devices, but it's hard to improve upon what currently exists for all locations. Like if there's a manhole in the middle of an intersection, it's not totally clear how a robot's going to make that better. Like someone's still going to have to go out there to retrieve a bottle probably. But I do expect, yeah, there will be some nice innovations in making that sample collection more automated and also less expensive.
Excellent. Well, thank you so much. I'm just going to thank everyone for being here. Thank you so much from Science at Cal. Thank you, Kara. Thank you, our audience. Very engaged, amazing questions coming through. So thank you so much. Again, we will have this lecture posted on our YouTube channel. We will send it out to everybody so you can share it with all of your friends. This has been such an insightful and educational talk. So I want to thank you, and engaging and totally engaging. You're getting so many thanks from our audience in the chat, Kara. So you should check those out. Thank you again. Visit us at science@cal.berkeley.edu. Goodbye, everyone. Have an amazing evening and see you next time.
Everyone, thank you all.